This fructan test kit, K-FRUC, allows for the specific measurement and analysis of fructan in plant extracts, animal feed and food products, which also contains starch, sucrose and other sugars. This method employs ultra-pure recombinant enzymes and specifically measures fructans, including inulin-type fructans from chicory, dahlia, Jerusalem artichoke and highly branched fructans from onion, wheat stems and leaves. The inclusion of endoleavenase has extended the use of this method to the measurement of leaven-type fructans, which are present in pasture grasses such as Timothy grass and are relevant to animal nutrition. This assay kit is used in 3 validated methods for the determination of fructan: AOAC method 999.03, fructan in foods, AOAC method 2016.14, fructans in infant formula and adult nutritional products, and AOAC method 2018.07, fructans in animal feed. This video tutorial will demonstrate the use of the kit with the supplied inulin control, the supplied leaven control and the sucrose control. The procedure shown in the video follows the protocol described in AOAC 2018.07, which is equivalent to the one in AOAC 999.03. The kit contains sufficient reagents for 100 assays, and a detailed assay protocol is available on the Megazyme website. The principle of the fructan method is shown in this slide. Fructan occurs in food and plant materials in the presence of sucrose and starch. For accurate measurement of fructan, the first thing that must be achieved is the removal of the sucrose and starch. This is achieved by enzymatic hydrolysis. The starch is hydrolyzed to glucose using beta-amylase, polulinase and maltase, and sucrose is hydrolyzed to glucose and fructose by a specific sucrase which has no action on fructooligosaccharides. These enzymes have been pre-mixed in the appropriate dosage in one of the kit components. They are ultra-pure recombinant enzymes and have no action on the fructan in the sample to allow for its quantitative analysis. After the enzyme hydrolysis step to remove sucrose and starch, the sample containing fructose, glucose and fructan is treated with sodium borohydride, which converts all the sugars, including the fructose and the glucose, but not fructan, to sugar alcohols. The solution is neutralized with acetic acid and adjusted to approximately pH 4.5. The mixture containing fructan and non-reducing sugar alcohols is treated with a mix of the enzymes exoinulinase, endoinulinase and endoleavenase, which have been formulated in one of the kit components to completely hydrolyze all fructans in the sample into fructose and glucose. D-fructose and D-glucose derived from the enzyme hydrolysis of fructan are then measured using the PABA reducing sugar method to give the fructan content. While the K-FRUC procedure is suitable to measure fructans, K-FRUC is not recommended for the measurement of commercial fructooligosaccharides and partially degraded fructan samples as shown in the next slide. In the commercial production of FOS, inulin is partially hydrolyzed to fructooligosaccharides with endoinulinase to produce oligosaccharides with reducing sugars, as shown in this figure. These reducing sugars react during the alkaline borohydride step and are therefore unaccounted for following the second enzyme hydrolysis step. Typically, this will lead to an underestimation of fructan content of commercial FOS of between 15 and 20%. This underestimation can be determined and allowed for if samples of the original fructooligosaccharides are available for analysis and standardization. Prior to sample analysis, prepare the required additional reagents as described in the kit assay protocol. Subsequently, once all of the additional reagents have been made, the kit components should be prepared, and once prepared, they are ready for use in the assay procedure. Bottles 3, 4, 5 and 6 are used as supplied. Dissolve the contents of bottle 1 sucrase enzyme mixture in 22 ml of buffer 1, 100 mmol sodium maleate buffer pH 6.5 containing 0.5 mg per milliliter of BSA by adding an initial 10 ml of maleate buffer containing BSA to bottle 1. Mix thoroughly and transfer this initial 10 ml of enzyme solution to a 30 ml polypropylene tube. Continue to rinse the vial with the remaining 12 ml of buffer, mixing thoroughly to ensure that all of the enzyme is transferred to the 30 ml polypropylene tube. This is termed enzyme solution A. Divide this solution into appropriately sized aliquots in polypropylene tubes and store below -10 degrees Celsius. Dissolve the contents of bottle 2 fructanase enzyme mixture in 22 ml of buffer 2, 100 mmol sodium acetate buffer pH 4.5 by adding an initial 10 ml of acetate buffer to bottle 2. Mix thoroughly and transfer this initial 10 ml of enzyme solution to 30 ml polypropylene tube. Continue to rinse the vial with the remaining 12 ml of buffer, mixing thoroughly to ensure that all of the enzyme is transferred to the 30 ml polypropylene tube. This is enzyme solution B. Divide this solution into appropriately sized aliquots in polypropylene tubes and store below -10 degrees Celsius. Add 10 grams of para-hydroxybenzoic acid hydrazide, PABA, to 60 ml of distilled water in a 250 ml beaker on a magnetic stir. Stir the slurry and add 10 ml of concentrated hydrochloric acid. Allow this to stir until the PABA completely dissolves. Transfer the solution to a graduated cylinder and adjust the volume of the solution to 200 ml with distilled water. Transfer the solution to a labelled Durand bottle and store at room temperature. This solution is stable for 2 years at room temperature. Prepare PABA solution B by adding 24.9 grams of trisodium citrate dihydrate to 500 ml of distilled water and stir to dissolve. Add 2.2 grams of calcium chloride dihydrate and dissolve by stirring. Add 40 grams of sodium hydroxide and dissolve with stirring. The solution may be milky but will clarify when diluted further. Add approximately 1 litre of distilled water to the beaker and allow the contents to dissolve by stirring thoroughly on a magnetic stirrer. Transfer this solution to a 2 litre graduated cylinder. Wash out the beaker with distilled water and also add the washings to the graduated cylinder. Adjust the volume to 2 litres with distilled water. Transfer this solution to a 2 litre Durand bottle. This solution is stable for approximately 2 years at room temperature. Prepare PABA working reagent immediately before use by adding 180 ml of PABA solution B to a 300 ml beaker. Then add 20 ml of PABA solution A and mix thoroughly. Transfer to a 500 ml Durand bottle. The mixed solution is stable for approximately 4 hours on ice. To prepare the alkaline borohydrate solution, weigh 50 mg of sodium borohydride into a 13 ml polypropylene tube. Add 5 ml of 50 mmol sodium hydroxide and mix thoroughly. Use this solution soon after preparation. This solution is reagent 3 and is stable for 4 to 5 hours at room temperature. In this Fructan analytical procedure, it is essential that the sample is milled to pass a 0.5 mm screen. Ensure the sample is mixed well by stirring with the spatula prior to sampling, and all samples should be at room temperature prior to being weighed. In this video, we are using the supplied inulin control in duplicate, the supplied leave-in control in duplicate, and the supplied sucrose control in duplicate. The sucrose control is assayed to show that the sucrase treatment step is effective. The value for this should be less than 0.2% weight per weight fructan. If the sucrase step has not been effective, the determined value will reflect the sucrose content of this control, which is approximately 10% weight per weight. The sucrose control should be analysed with each new lot of reagents. For samples containing 0 to 10% fructan, like the leaven and sucrose control, accurately weigh 400 mg of the sample into a dry Pyrex screw cap culture tube, dimensions of 25 by 150 mm. For samples containing 10 to 40% fructan, like the inulin control, accurately weigh 100 mg of the sample into a dry Pyrex screw cap culture tube, dimensions of 25 by 150 mm. Add 25 ml of distilled water to the sample tubes and mix thoroughly. Ensure all of the sample has been manipulated back into the bottom of the tubes, as shown on the vortex. Loosely cap the tubes and place in a boiling water bath for a total of 10 minutes. After 5 minutes, tighten the tube caps and vigorously mix the contents by vortex, ensuring again that all sample has been manipulated back into the bottom of the tube. Return the tubes to the boiling water bath. After a further 5 minutes, remove the tubes from the boiling water bath and again mix thoroughly by vortex. Allow the tube contents to cool to room temperature. Vortex the samples and transfer approximately 2 ml aliquots of the samples to labelled 2 ml microfuge tubes. And centrifuge at 13,000 RPM for 5 minutes. For samples that have greater than 40% weight per weight fructan, after centrifugation add 1 ml of the centrifuged extract to 2 ml of distilled water and mix thoroughly. Use the supernatant or the diluted supernatant and apply to the removal of sucrose, starch and reducing sugars procedure. Accurately dispense 0.2 ml of each of the solutions to be analysed, which contain between 0.1 and 1 mg per milliliter of fructan, directly into the bottom of a glass test tube of 16 by 100 mm dimensions. Add 0.2 ml of diluted sucrase amylase solution, enzyme solution A, directly to the bottom of the sample tubes. Mix the contents thoroughly on a vortex mixer. And incubate the tubes at 30 degrees Celsius for 30 minutes to hydrolyze the starch, sucrose and maltose completely to fructose and glucose. After 30 minutes, add 0.2 ml of reagent 3, alkaline borohydride solution, to each tube in a fume hood. Cover the tubes with parafilm and stir the tubes vigorously on a vortex mixer and incubate at 40 degrees Celsius for 30 minutes. This will ensure complete reduction of reducing sugars to sugar alcohols. After 30 minutes, add 0.5 ml of 200 millimolar acetic acid to the tubes in a fume hood. Mix vigorously on a vortex mixer. A vigorous effervescence should be observed. This treatment removes excess borohydride and adjusts the pH to approximately 4.5. This is solution S. For health and safety reasons, it is important that no naked flames, for example Bunsen burners, are present in the lab near solution S, and also that the tubes containing solution S are not closed tightly, as overpressurization may occur. Accurately and carefully transfer 0.2 ml aliquots of solution S for each sample to the bottom of 3 16 by 100 mm glass test tubes. These test tubes serve as the sample in duplicate and the sample blank and should be labelled accordingly. It is essential that the sample aliquots are delivered directly to the bottom of the tubes to ensure that the sample will come in complete contact with the next enzyme added, which is the fructanase solution. Add 0.1 ml of the fructanase solution, enzyme solution B, to 2 of the tubes for each of the samples. To the third tube for each sample, add 0.1 ml of 100 millimolar sodium acetate buffer, pH 4.5. This is the sample blank. Seal each of the tubes with parafilm. Stir the contents on a vortex mixer and incubate the tubes at 40 degrees Celsius for 30 minutes to allow for the complete hydrolysis of fructan to defructose and deglucose. With each set of determinations, reagent blanks and fructose controls must be included and analysed concurrently. During the 30-minute sample hydrolysis incubation, prepare the reagent blanks by adding 0.3 ml of 100 millimolar sodium acetate buffer, pH 4.5, to label tubes in duplicate. Prepare the fructose standard solutions by adding 0.2 ml of defructose standard solution, kit bottle 6 at 1.5 mg per milliliter, to 0.9 ml of 100 millimolar sodium acetate buffer, pH 4.5, and mix thoroughly. Dispense 0.2 ml aliquots of this prepared fructose solution, containing 54.5 micrograms of defructose, to label tubes in quadruplicate. Add 0.1 ml of 100 millimolar sodium acetate buffer to each standard tube. Remove the tubes from the 40 degrees Celsius water bath and also remove the parafilm from each tube. Add 5 ml of PABA working reagent to all tubes, the samples, the sample blanks, the fructose standards and the reagent blanks. Mix the tube contents thoroughly on a vortex mixer. Incubate all tubes in a boiling water bath for exactly 6 minutes. This incubation duration is critical and should be carefully timed. After exactly 6 minutes, remove the tubes from the water bath and immediately place into a cold or ice water bath for approximately 5 minutes. Measure the absorbance of all solutions at 410 nanometers against the reagent blank. This absorbance measurement should be performed as soon as possible after cooling the tubes. The absorbance readings of the sample, standards and the blank reactions are used to calculate the fructan content of the test samples on an as-is basis. The calculations of fructan can be performed manually as described in the calculation section of the assay protocol. Alternatively, we have also developed specific Excel-based Megacalc applications for each kit of the Megazyme range to allow quick and easy results analysis. Results can be analysed using the Megacalc application specific to this Fructan Assay kit, which is available to download free of charge from the Megazyme website. The Megacalc spreadsheet provides full instructions for use. Open the Megacalc worksheet and input the following: sample details, absorbance readings for the D-fructose standards. For each sample, input the sample identifier. For each sample, input the blank absorbance values. For each sample, input the sample absorbance values recorded in duplicate. Input the recorded sample weight used. If dilution of the sample has been performed, then input the dilution factor used. If samples are not diluted, for example the samples containing up to 40% weight per weight fructan, the dilution factor is 1. For samples containing 40 to 100% fructan, these should be diluted threefold after the extraction and centrifugation step, and therefore, for these samples, the dilution factor is 3. When all of the data has been entered, the content of fructan in the sample is automatically calculated and given as grams per 100 gram on an as-is basis. If the moisture content of the sample is known, enter this percentage into the moisture content column. Subsequently, the fructan content of the sample will be calculated and given as grams per 100 gram on a dry weight basis. For further information, please see the paper on the measurement of total fructan in foods by enzymatic or spectrophotometric method, a collaborative study by McCleary et al., which can be accessed from the Megazyme website.